feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, and welcome to the National University of Singapore Yonglulin School of Medicine Healthy Longevity Webinar. We have a very interesting talk tonight from Dr. Daniel Stickler, who will be telling us about longevity medicine in practice. I'm sure there'll be a lot of questions about this, so I'd like to remind you to use the Q&A function to send questions, and we'll try to get to as many of them as possible. Before we start with Daniel, we have Li Hui-Chi, a PhD student at the Center for Healthy Longevity here at NUS, and she will talk about her own study about an association between sleep duration from midlife to late life and the risk of depressive symptoms, the Singapore Chinese Health Study.
Thank you for your kind introduction. As mentioned, I'm Hui Qi, a PhD student from Prof. Kong's team at the National University of Singapore. Today, I will be presenting one topic of my projects, which is about the changes of sleep duration and late life depression. With the world aging rapidly, older people are increasingly facing mental health challenges, and among them, depressive symptoms are quite common problems. It's estimated that the prevalence of late-life depressive symptoms ranges from 17% to 34% worldwide, and 8 to 10% of them transition into major depression every year. Although the pathophysiology of late-life depression is unclear, research has shown that oxidative stress and brain inflammation may cause neurodegeneration leading to depression. Therefore, modifiable factors, such as sleep habits, could help prevent late-life depression by targeting oxidative stress and inflammation. Therefore, we aim to investigate the association of sleep duration at midlife and subsequent changes over 20 years with the risk of depressive symptoms in late life. We use data from the Singapore Chinese Health Study, which is a population-based cohort study. But for the present project, we included about 14,000 middle-aged participants who completed all baseline and follow-up visits over 20 years, with assessment of every 5 to 6 years. Sleep duration was assessed at baseline, follow-up 2, and follow-up 3, and change in sleep duration was defined as a difference of equal and more than 2 hours between baseline and follow-up 2. While our outcome, the depressive symptoms, were only measured at follow-up 3 interviews, using 15-item geriatric depression scale, and 23.8% of our participants had depressive symptoms. Modified persona regression models were performed, with adjustment for potential confounders. Here are the results. For the prospective analysis at baseline and follow-up 2, we found only short sleep duration was associated with risk of depression. In contrast, for the analysis at follow-up 3, where sleep data and the depressive symptoms were measured at the same time, we found both short and long sleep duration were associated with high risk of depression. Hence, we consider short sleep duration could be both a risk factor and a symptom of late-life depression, while long sleep duration only concurred with as a symptom. Then we further examine the changes of sleep duration. For those with recommended sleep duration of 6 to 8 hours at baseline, About 63% of them maintained their sleep duration at follow-up 2. Others either decreased or increased their sleep duration at follow-up 2, and we found a risk for those who decreased their sleep duration. For participants with short sleep duration at baseline, regardless of whether they maintained or increased their sleep duration at follow-up 2, they all had a high risk of depression. Even they prolonged their sleep at follow-up 2 is still harmful. On the other hand, we found evidence of no risk for those with long sleep duration at baseline, regardless of whether they changed their sleep duration or not. We consider the possible mechanisms are that chronic sleep deprivation is linked to increased inflammatory cytokines and dysregulated neurotransmitters, both associated with depression development. Animal studies also show that sleep deprivation may lead to neurobiological and neuroendocrine alterations. But surprisingly, these changes were reversible in young mice, but not in old mice, likely due to the reduced brain plasticity with aging. This also supports our findings in human studies that sleep lengthening after midlife couldn't reduce the depression risk associated with short sleep duration at midlife. In conclusion, we found short sleep duration at midlife and later was associated with a high risk of depression in late life, and this risk was not reduced by subsequent prolongation of sleep. Hence, the promotion of sleep hygiene may have to happen at early life to reduce the risk of depression and improve quality of life among older adults. I would like to thank my supervisor, Prof. Kong, and our research team for their support and guidance. Thank you for your attention.
Thank you. So make sure and get your sleep. Not something I'm always very good at. Tonight we have Dr. Daniel Stickler. As the co-founder of Apeiron Centre, he stands at the forefront of systems-based optimization and age rejuvenation medicine, a transformative approach that transcends the conventional symptom-based reductionist framework in favor of a comprehensive systems science perspective. His clinical practice is at the vanguard of pioneering clinical trials sculpting the future of age rejuvenation and human enhancement. His talk tonight is going to be about longevity interventions in clinical practice. I've known Daniel for a few years now, and it's always fun talking to him about what's going on because he really is at the forefront of trying different interventions. So I'm excited to hear the talk tonight, Daniel.
Thanks for the intro, Brian. Yeah, I love having the conversations between the clinical side and the research side. We always come up with some pretty unique ideas. I'm going to share my screen here and talk about what we're actually doing in the clinical practice that's available right now for aging interventions. And one thing we do differently is we take a very systems-based approach. We've been doing this for about the past 6 or so years, where we're looking at many biomarkers in our clients. So we get molecular markers, biochemical, genetic, epigenetic, transcriptomic, metabolomic, proteomic, environmental, psychological, social, even spiritual. And this is a way to consider the systems-based approach. This was the Foresight Institute's obesity map that they did back in, I think it was 2007. And you can see all of the interactions of the different areas that contribute to the core thing that they were looking at, which was energy balance. This was a beautiful map, and it was designed to disrupt the obesity epidemic in the UK, but policymakers couldn't understand it very well. And that's been one of the issues with systems approaches. So the way we've been doing it, we get full genomic sequencing now. We get brain EEGs. We assess the autonomic nervous system through HRV, breath mechanics, expired CO2, posture and gait analysis, microbiomes, environmental exposures. And what we try to do is create context for each of our clients. We get quality of life inventories, ethical and spiritual values, geopolitical leanings, meaning and purpose in their life. We do the cancer screens, we do skin and retinal scanning, glycans, epigenetics, metabolomics, proteomics, transcriptomics, full body MRIs. And recently we've added things like the coronary CT angiogram with Clearly analysis to look at soft plaques. And we're working on an IRB with Caristo Health out of London for the Carry Heart, which looks at inflammation around the blood vessels from this same scan that Clearly uses and the same one that you get with a coronary CT angiogram. We're working on an IRB with Sage Signal, which is an at-home test that will determine inflammation levels in the brain. Sapir X, a new one that I love, and I think Natalia has been on the show talking about it, looking at cellular and immune senescence tests. And one of the things we're finding is that a lot of the biologic age tests that use surrogates, they use proxies, they're coming back very different from the immune aging that we're seeing with Sapir X, which is more direct tests. So we're interested in how that compares. And we're collecting wearable data, the biometrics, and keeping all of this data in a large database. So I'm going to transition. I wanted to give you that background into the way we approach things, but I want to talk about the therapies that we've been doing in the clinic. Some of these here in the United States, some of these in a special economic zone off of Honduras on an island called Roatan, which is fast growing in the longevity space. We do plasma dilution therapies. We do young plasma exchange. This is using 18 to 24-year-olds to replace plasma in individuals. We're doing plasma gene therapies using follistatin and getting ready to start doing clotho. We're involved in some early access programs from the FDA with galectin-3 antibodies. And so let's talk about some of these. The old plasma, I mean, we're talking about age-accelerating chronokines in there. We're talking about inflammatory proteins, SASPs, cellular waste products, misfolded proteins, and other progerotic factors. And the Convoy labs at Stanford did most of the early research on this and still continue to do that. But in their paper, they found the results of biological age are strongly supported by the data, demonstrates that rounds of therapeutic plasma exchange produce global shifts to a younger systemic proteome. And this has been fairly consistent. I mean, young plasma is fairly well established as a bit of a, I won't say youthening, but rejuvenating for sure approach. And when we talk about this, you know, we have in the old and young, we have in the old progerotic factors, and in the young, we have youth factors. So there's a big change in what we're seeing in the plasma in these two groups. And so that's what prompted really looking at, okay, well, what if we take off all of these progerotic factors in the plasma, but then we replace it with plasma from a young person, specifically the 18 to 24-year-olds in the study we're involved in. And the reason for that is those are individuals that tend to donate blood to plasma centers. And it's almost like they're at the peak of their physiologic health. This is a point where they're at the peak reproductive years. So all of their different substances in the plasma are really generating peak health in the individual. There was a lot of interest in this when Catcher published studies talking about his E5, where he was injecting old rats with this magical substance called E5. Turns out this was basically exosomes from the young plasma, and it was actually porcine exosomes, young porcine, and it was rejuvenating these rats. And this is essentially what we're doing with the young plasma. So the composition of young, fresh frozen plasma that we get from the 18 to 24-year-olds, there's 1.86 billion exosomes in just 1 milliliter of this stuff. And we're replacing a full liter and sometimes 2 liters in some people. A full liter, you're talking 1.86 trillion exosomes. I mean, this is enormous. But on top of that, you're also getting these supporting factors. So it's one thing to give the exosomes, but I think Tony Wiscore's lab talked about the need for some of these support factors that are going to be in the blood of younger species, where you need these proteins, the peptides, cytokines, sex hormones, enzymes, and minerals that will support that more youthful phenotype that you're trying to get with the exosomes that are being infused. And this was another paper from Wiscore's lab. Intriguingly, we observed an almost universal loss of gene expression with age. Aged blood reduces global gene expression, and young blood restores it. And I think he was working with the brain capillary endothelial cells with this, and they noticed profound changes with the young blood when this was transfused. Now, I want to talk a little bit about the plasmid gene therapies that we're involved in, and these are early studies in this. This is a company that's working with plasmids, so they're taking E. coli, making the proteins, and specifically the follistatin protein, the follistatin 344, and they're putting this plasmid with a delivery vector that is not viral. It's a PEI delivery vector and is a cationic polymer and does not create an inflammatory response in the body, but delivers these plasmids to the cells and gets them into the nucleus, and it does not incorporate into the DNA, it was a real plus when they came to me and told me about this. I held off on participating in it until they came back recently with a kill switch, where you can take 3 days of tetracycline and it completely eliminates the plasmid. So it was a safety feature that I really liked. And in animal studies, they found with these plasmid therapies, you get increased muscle mass, 32.5% increased lifespan in mice. The early human studies showed excellent safety profile with this. And they also noticed targeted reduction of visceral adipose tissue and decrease in chronic inflammation. I actually took this plasmid therapy a little over a year ago. And my results at 6 months were pretty impressive. And I was a good responder to it. My lean mass went from 117 pounds to 123. So basically about a 3 kilo increase. Fat mass dropped 9 pounds. Visceral abdominal fat, it's interesting. The visceral abdominal fat seems to be a really high target with the follistatin. Body fat percentage dropped from 22.8 to 17.8. And my average HRV increased by 22, which was one of the most impressive things that I found because I do monitor HRV very closely. And we're getting ready to do some trials with clothoplasmid. And, this is a bit of a holy grail for me because I feel like clotho protein, when in that secretion state where it's a continuous low dose secretion of it, it's been associated with higher general intelligence, enhanced synaptic plasticity, neural resilience, arterial calcifications, longer lifespan, all of these things that are so great. I mean, even improving kidney function and heart function. So this is one we're excited about and currently still in the lab with it, but hoping to get this into a clinical trial very soon. Another one I want to mention is the anti-galectin-3 antibody. This is FDA has an early access program, so we're working with this in our clinic in the U.S. Using the TB006 for dementia. And this is it's an interesting approach because you're talking about the beta amyloid monomers, forms dimers, trimers, oligomers, and the galectin-3 seems to be weaving this together and holding it in place. The TB006, which is the anti-galectin-3 antibody, goes in and breaks up the galectin-3 oligomers and allows for the clearance of the plaques. And the early studies looked really good, great safety profiles. So the FDA approved an early access program for certain clinics to perform this. And we currently only have one person in the trial, but we're seeing some cognitive improvement. He's had about 5 monthly over 5 months, he does a 1 month IV of this. But you look at galectin-3 and it's kind of ubiquitous in all areas of the body. I mean, it's involved in all of these brain conditions, Alzheimer's, Parkinson's, TBI, long COVID, inflammation in the heart, inflammation, congestive heart failure, myocardial ischemia. It's involved in oncogenesis, fibrosis in the lungs, and in neuropathies, and in aging, senescence, immune dysregulation, tissue fibrosis. I bring this up because this brings us full circle back to what I was talking about at the beginning of looking at things from a systems approach. It's really easy to look at this and say, wow, this is an incredible intervention. I mean, if it can affect all of these different programs. But when you look at it from a systems approach, you have to consider not only these positive impacts on certain systems, but also the consequences to these systems, because galectin-3 is ubiquitous and it is involved in natural healing processes. It's involved, like after a heart attack, it causes more fibrosis, but blocking it may not be the right thing to do after a heart attack. So we're not really sure of the nuances of this and how will using it to treat the brain impact other areas of the body in this systems-based approach of these individuals. So it's real easy to latch on to all of these positive findings with this. I actually saw a recent study that talked about clotho, that there was a bit of a U-shaped curve on cancers. Low clotho had higher levels of cancer rates. And then there was this nadir and then a slight increase as clotho levels went higher and higher. So there's a lot that needs to be considered when we're looking at this. And, you know, I'm not a huge fan of the population-based studies because they haven't done a great deal for us over the years. I mean, you look at the top 10 prescribed drugs in the US, and I think you have to treat a minimum of 4 people to get one positive effect at the best of these top 10 prescribed drugs. And there just is not enough contextualization of how these interventions work on individuals. And so that's something that that I have wanted to address for quite a while. So I've given a lot of information here, and I wanted to leave some time, Brian, for us to have some discussions on this.
No, I think we can manage to handle that. Let me start with something that struck my attention. I see, what gets measured when people come into clinics, and I've never seen geopolitical leanings before. I'm wondering, maybe I'll ask a question in a way that won't get you shot in Texas. I'm wondering if um, the strength of geopolitical leanings are associated with anything, not necessarily which direction you lean, but how strongly you feel about, looking at a very polarized country. This is a very interesting place to maybe ask questions like that in terms of health and mental status. Is there any association you see there?
We haven't yet. I mean, we're collecting this data because we feel like there can be associations here. And one of the really interesting things about what we've discovered on systems is we try not to classify the data points too much. We try not to guide the AI to find associations that we think are going to be there. I mean, it seems to work best with this chaotic distribution of data points and allowing the AI to start finding patterns and associations without us putting our bias on it too much.
Yeah, I don't know how many people you have, but I think one of the things when I'm talking to clinics is that we have to get past this like old style statistics of comparing one thing to one other thing and really start to think about how to model individuals in terms of their baseline status, in terms of what interventions they're doing over time, and in terms of outcomes, because I think that you probably need a lot of people for this, but once you get to a certain point, you'll start to find trajectories of response related to complex baseline, starting points. And I don't know how you feel about that, but that's something that we're trying to think about how to do statistically now.
Yeah, I mean, it's gonna take a lot of time and a lot of enrollment for sure, but I feel really strongly that this is going to create predictive models. I mean, it can take an individual and say, based on this information, your trajectory is this, and if you do this, we may be able to disrupt that outcome.
So this say at the state we are right now, a lot of people are promising personalized approaches to longevity or precision approaches. Are we there yet?
We're getting closer. The rapid improvements in AI is what I think is going to take, because it was just like that foresight map, obesity map. I mean, it was a beautiful map, and if people understood it, they would have been able to really make huge headways into obesity. But it's just very hard. I mean, complexity thinking is a whole different way of approaching problems, and the AI can make it so much easier because it can analyze so many data points simultaneously that we just can't do with the human brain.
Yeah, I think a lot of people now are talking about meaningful AI, and meaning that whatever the AI spits out can be interpreted back in terms of mechanisms or understanding about pathways and what's going on. I don't know how you feel about it. Is AI going to get there for this kind of use or not?
I think so. I mean, with the advances in probabilistic computing, I think we're getting closer to getting rid of these hallucinations and finding out why some of this stuff is occurring. Right now, I think it's important for us to start collecting all the data, get all the data together, and within 2 to 5 years, I think we'll have the computing capacity to analyze this data without us putting too much of our own biases into it.
Yeah, that's really something that I'm encouraging clinics to do. And I've been joking that billionaires are the new model organism, and or multimillionaires anyway. And so it's great if clinics are willing to, maybe share or at least analyze the data and report some of the findings, because some of these experiments, I know you're doing them through clinical studies too, but some of these things you're doing are not things that are easy to do in an academic setting. And so the data that's being collected is um, extremely important, I think, to understand in terms of longevity. I think everything you mentioned tonight has a lot of promise, but I don't think we know yet what's going to work and what's not going to work and who it's going to work in. And so, in clinics out there listening, please analyze data. I guess it's I'm begging rather than asking a question here, but I think it's so important.
Well, that's an important point, because I mean, most of the longevity clinics are doling out the rapamycin pretty regularly with their clients, but there are some clients that you have to do different doses with, and there's clients that you need to avoid doing it with.
Mm-hmm.
And getting the data, I mean, right now, labs in the U.S. can analyze sirolimus levels, and if you get it a couple hours after your dose, you can kind of get an estimate of the serum half-life and levels. And then you have tests like the Sapir-X, where you're looking at the senescence of the immune system and the resiliency of the immune system as well.
Yeah. I really like that test too. I haven't seen a large amount of data with Sapir-X yet, but for one thing, I did really well in that test. So those are the only people I let on the show. If I do really well in their biologic aging test, then they can come on the show. No, I'm joking. I think that's a really exciting test though, because it really, it measures aging at the same time. It does in a way that makes mechanistic sense, or at least it addresses some of the known pathways linked to aging. So you get more information than just the numbers. So I think that's, and I think they also try to interpret it in a very meaningful, well thought out way, as opposed to just spitting a number back. I haven't seen a large amount of data yet, so I can't speak to that, but I think the concept is really cool. So I wanted to ask about the plasma therapy a little bit. Um, and so the question I've always had is, I think you made a great point. There's a lot in this young plasma, but how frequently do you think it needs to be done to have an effect? And what effect do people see when they try to do this? What's the most common thing that you believe happens?
Yeah, and this is mostly going to be anecdotal. We just don't have a large volume.
I understand.
We've gone through that. And we're trying to figure out what the ideal frequency is. I think with with apheresis itself, we're looking at 3 to 6 months, and it seems the effects that we see seem to persist a minimum of 3 months after an apheresis. And there's some residual effects at 6 months as well. The young plasma, it seems to be a little more profound. I mean, we see reductions in LDL-P levels of the cholesterol. We see reductions in small LDL-Ps with both apheresis and young plasma. It seems to be a bit stronger with the young plasma in that regard. We're seeing significant reductions in inflammation in the body with both procedures. We seem to find that the subjective well-being is better with the young plasma, though. That one, definitely we see improvements in HRV, sleep patterns, that are more so than what we're seeing with just the apheresis alone.
So it sounds like from your statement, there was some thought or some hypotheses lately that it's mainly about diluting the bad factors from old blood or old plasma that's giving the benefit. But I think you're making the case that there are actually many things in the young plasma that are having a positive effect as well as just diluting things.
Yeah, and that's our theory on that, based on the physiology of it, but it has not. We don't have enough data to prove anything on that quite yet.
Yeah, I think that's a fascinating question. And then beyond that, what are those factors? Because you list a lot of potential things that could be beneficial, and I still think there's a lot of research that needs to be done to understand which of those things are actually providing that benefit. But um, I think one of the most interesting things you talked about was the gene therapy because not many people have tried to do that yet. And the plasmid approach with the kill switch is very interesting. Um, how long? What's the longest that people have done it? And I'm just curious. Like, you see these changes in lean mass and fat mass. Do they happen in the first 3 months and then stop, or do they just continue to happen, or what's the persistence of this effect? I guess I'm going to keep asking that question with every intervention tonight. Sorry.
Yeah, so the long they've done this injection for about 4 years now, and they initially started in injecting it into the subcutaneous fat cells, and those cells turn over a little bit more rapidly than we see with the muscle. So they were getting about 2 years before it kind of burned out in those tissues. Recently, the injections have been into the muscle, and the hope is, maybe 3 to 5 years of benefit from it, and then you just do another injection. But it's pretty amazing how well they've figured out the dosages of this. I mean, it's very consistent with the dosages that people get that bring them into that youthful level of the follistatin, and having that continuous flow of it seems to be ideal. Now, it does seem to occur in the first 3 to 6 months, the bulk of the effect for sure. Especially one of the most profound things is everybody raves about their recovery from exercise. It's a completely new and improved recovery that they're seeing with their exercise. The fat loss, I've actually seen a couple people that had to turn it off because they were losing too much body fat. They started losing the body fat and then tried to eat enough calories to keep the fat on, and it wasn't working very well. So had to have their plasma turned off.
And the kill switch worked in that context?
Yeah, the kill switch study, I was actually a principal investigator on that, and it was, it was very good. It was amazing how well it did, and they're going to be publishing that soon.
Cool. You mentioned that you talked about your data, and you mentioned that you were one of the responders. I don't know how much you can talk about other data, so I don't want to put you in a bad position, but what's the frequency of responders to this? If
Well,
you
no,
can answer that question.
I would say I was a hyper responder. Pretty much everybody responded. And a lot of these in the safety studies. So, the data that we have was more of the safety studies.
Yeah.
So it wasn't a study to determine that kind of outcome. They were people from like the age of 25 to 85 in that range. And older people definitely responded significantly more so than the younger. The younger group noticed more of the recovery response and less of the fat loss. And the muscle gain was pretty decent in that group. But most of them didn't. They weren't actively doing anything. Some of them weren't exercisers. Some of them were just eating a random diet. And it was just to get the safety profile of this. So I think when we get to studies that really look at what it can do for somebody taking a positive approach to health, I think it's going to be more profound in that group.
I think it also addresses a question that I think is important when it comes to recruiting people into studies, which is that sometimes when they say do take our longevity approach and in 15 years from now you're going to still be healthy. I think that it might motivate people, but it's hard to keep people motivated by that. So I think the approach that in my experience seems to work better is, we're going to improve your performance now and protect you later. I would think that something like this follistatin therapy has a performance outcome in the near term as well as hopefully protection in the long term as well. So it fits that category very well, I think. So it's great to hear that. I'm assuming the safety data is good, or you wouldn't be talking about it. So it's good to hear. It's good to hear that. So,
Yeah, I mean, everybody's wanting that magic bullet. I mean, in the clinical practice, people come in and they're wanting a magic bullet. And, I tell them 75, 80% of health span is determined by lifestyle. And if they're not getting good sleep, if they're not eating well, and exercise, I think exercise is number one for sure. If they're not doing these things, any of these interventions right now are just not going to work very well.
yeah, although, I still believe that and would have said that more strongly. But, as you mentioned, the response was better in older people. And we saw that also with AKG, that in the clinical study we did, the response was better in biologically older people or chronologically older people. And so I'm wondering, I think we need to do this with a wide range of longevity interventions because some things might only have an effect in people that are exercising, and other things might only compensate for lack of exercise. And I think to use exercise as an example. So I think we're all, I think what you're saying, I generally agree with, but we're all kind of guessing which interventions are going to help, which people and how they're aging. And so I think that'll be stratifying results is going to be fascinating over the next few years.
Yeah, and that brings us right back to the contextualization of each individual.
Yeah. I also, you put me in touch with True Binding. I want to have them on at some point too on the show. I think their data on AD, is quite good. And so this antibody therapy. So my first instinct when you talked about it and when they talked about it is it would be good for longevity because of these effects.
Yeah.
But, you also mentioned that you need that protein around doing things at certain times. You could argue for that with Tor too. So a lot of these interventions like this or rapamycin or others, it might be about delivering them intermittently at the right time and not at the wrong times ultimately to get the biggest impact. But my guess is if you did a longevity study with that antibody, you'd see a positive effect.
Mine too. Yeah.
Yeah. Maybe before I go to the audience, I'll ask, just talk to me a little bit about Roatan. We haven't talked about that on the show. And that's an interesting concept in trying to accelerate potential beneficial interventions, both therapeutically and in terms of healthy longevity. And it relates to regulatory zones. And maybe if you don't mind, can you give your perception of that?
Yeah, it was a new concept to me as of about a year and a half ago, but around the world, they're setting up these special economic zones. So they are zones that basically are creating rules of government for themselves in these zones. Prospera, which is on Roatan, is a special economic zone that crypto and longevity are the key focus there. And there's several longevity startups that are setting up shop down there. They have their own IRB. It's headed by a former deputy director of the FDA. So it's not like it's just a made up board to get approvals. But they fast track IRBs down there. They have teams that help you to create them. So it's just a facilitated process for, you know, in the U.S., there's such a backlog in trying to get IRBs approved, and startups in that they have a lot of difficulty breaking into that, and that's where a lot of the longevity research is being done right now, is in the private sector, and this is really helping them out. But then a lot of the scientific sector is actually tapping into some of these clinical trials. They're very interested in getting people into the trials, and there's a lot of people that they're willing to be early adopters. I mean, they're choosing to do this, and as long as you give them as much of the detail that and tell them what you don't know about it, I'm strongly in favor of letting people have that sovereignty to make that decision.
Yeah, I actually agree with that too. I think it's about transparency, but I think we've taken too many decisions out of the hands of the individual in terms of healthcare, and that's one of the reasons when you tell them to do things like make lifestyle changes or take drugs, that the compliance is not that high. Instead of educating people and getting them, engaged on board and accepting the approach in a sustained way. We're telling people what to do, and then we're surprised when they don't always listen. So I think that that concept is a very good one, and I think hopefully we can continue to get more decisions back in the hands of individuals. So I could ask questions all night, but Kalisha is getting bored waiting. So I think better I'll bring her in and she can ask some questions from the audience.
Okay.
Hi, Dr. Daniel. Thanks for the presentation. So we have a number of questions here from the audience. We'll start with the plasma transfusions. So Arthi asks, is it possible to stimulate the body to produce healthy plasma rather than the transfusion? What are the possibilities here? Because it probably would be quite hard to get so many plasma donors to serve a wider population of aging people.
Well, the big question is, what is healthy plasma? I mean, you saw how many different components are in that young plasma, and that's just the ones we know about. So I know there are companies right now that are working on an artificial plasma with some of the key factors that we know are in there, and with the way we're able to start really creating proteins so easily right now, I think that's a possibility in the near future for sure.
Okay, great. Is there a certain type of young plasma that gives a better outcome? Like if the donor had a healthy lifestyle or an active lifestyle, do you see better outcomes?
I'm sure you would. We don't get that kind of data on these donors, and these donors are usually college students. It's a plasma center we get it from in a college town. So they're mostly college students. They could be stressed, they could be out partying. You don't know really what you're getting, but in general, I think you're getting fairly healthy people.
I see. Okay, so assuming as people age, their immune system would react differently to the external stimulus or the plasmid therapy, would you suggest different types of therapies to people at different stages of their lives?
Well, that's what we're trying to determine with this contextualization. Is there a way to analyze one individual and say, well, in this person's context, this will tend to work better than somebody here. I mean, it's just even like looking at their genetics and drug response. That's contextual there. And I mean, I think that's what it's all going to come down to, is really looking at individual response. I mean, these N of 1 trials, I feel like are going to be the studies of the future.
Sounds really tough to study, but you'd probably be powering that with AI too, won't you?
Mm-hmm.
Mm-hmm. So
right.
Onto the follistatin therapy. Isu says it's really intriguing that the follistatin plasmid gene therapy increases muscle mass and decreases fat mass. Could you elaborate further on the underlying mechanism of this? And does the therapy affect fat metabolism?
I'm not sure what the response is in the fat. My understanding with the muscle is that it's countering the increase in myostatin. So as we age, our follistatin levels drop off, myostatin levels increase, and with the muscle, I know it's a big part of it is this inhibition of the myostatin.
Mm-hmm. So not so much studies on the effects on fat yet.
I don't know. I'm not that familiar with the mechanism with the fat cells.
Okay.
There's certainly a lot of interaction between muscle and fat too. And if you look, for instance, in mutations in mice that cause muscular dystrophy, at least in the laminin mutations, they're running out of fat basically because the muscle's ineffective and it's burning a lot of energy to try to be functional. So in this case, if you're creating more energy burning in the muscle or more muscle mass, that may just place a higher demand on this, energy substrates and reduce the fat in a secondary way. I mean, there may be more direct interactions too. So,
one aspect I'm curious about is, we're making good progress on sarcopenia from a hypertrophy standpoint, from a muscle mass standpoint, but most of our interventions aren't doing anything for the strength loss that's occurring even with the increased muscle mass. And I'm wanting to look at the follistatin to see if that's making a difference there.
Mm-hmm. We also have a question. What are your thoughts on mini-circles follistatin gene therapy approach? Do you think it has increased risk compared to traditional methods? And does it have broader applications in anti-aging and disease treatments?
Well, mini-circles, who I was talking about.
Oh, that is.
Yeah.
But there are other approaches that have been tried, right? Can you maybe describe?
I mean, gene editing, for one, is taking a viral vector and it's actually editing the native genome. And, here's another aspect of this, you've got to look at everything. I mean, in the initial studies with the Cas9, they were looking at what they were trying to do, and they were like, oh my God, it's perfect surgical replacement of this area of the genome. And so there was all this hype about it. And then they started pulling back the microscope a little bit, and suddenly they were finding other areas of the genome that were also being edited. So they didn't know that these other areas were involved. And so what happens with that? I mean, what's the consequence of that? And the improvements that they've had in the gene editing are profound. But I think there's a role for the gene editing in certain things. I prefer with enhancement or age rejuvenation approaches. I prefer the plasmid approach just for the fact that I'm not editing the genome there, and I have a way to turn it off.
All right. Speaking of the ways to turn it off, can you, apart from doing that kill switch thing, is there another way that you can control the expression level of the plasmid? And how long does the plasmid linger within the cell?
They're working on that right now. And you can take a lower dose of tetracycline and dial down the production. That's one study that they're working on right now. When you take the 2 doses of tetracycline over 3 days, we see a complete kill of the plasmids themselves. But yeah, it does seem like there is going to be a way to reduce it, not something to increase it, but to reduce it for sure.
Mm.
How much replication is going on in vivo with these plasmids? I mean, is it just you keep what you get, or are they actually replicating?
No, you just keep what you get. They're just utilizing the nuclear machinery to produce the protein, and like I said, they're pretty amazing how close they're getting to knowing how much is going to be produced with a certain amount of the injection.
Okay, great. We just got another question in about the plasmids. Is there any risk of the plasmid entering the nucleus and undergoing line 1 mediated reverse transcription and integrating into the genome?
I'm sure there is. I don't know the answer to that, but there's still a lot of unknowns in this. From what we've seen in the animal studies, it seems to be doing what they expected it to do, but it's just like the Cas9. Let's pull the microscope back and see what else is happening there. It's just a lot of unknowns still remain.
All right. So traditionally, a lot of bodybuilders just take follistatin peptide to increase muscle mass. What are the differences between taking the gene therapy or the plasmid follistatin compared to just the protein?
Yeah, that was one of the reasons that that Minicircle went that route, because of the continuous dose. The bolus dosing that the bodybuilders were doing were creating complications. I can't say exactly what those were, but they were not pleasant side effects that occurred from the bolus dosing of the follistatin.
Mm-hmm. Yep, and the plasmids don't have those side effects, of course.
They don't seem to. The safety profile was pretty significant. It was hardly any issues whatsoever.
A lot of this dosing with weightlifting is taking a very large amount of protein, and I don't know how much of it even gets absorbed into the bloodstream, but altering your protein composition dramatically can have some digestive complications that you don't want. So
Okay, we have a question from Dishani. They say, great to see a comprehensive approach to health. Dishani is a toxicologist, so they are curious about what you mentioned regarding environmental investigations. Do you measure environmental metabolites in biofluids? And if so, which ones?
We are working on that. We're trying to find a validated testing that we can do for a reasonable cost. We like the air quality monitors that are getting better and better right now. Just to be able to have an air quality monitor that's just like a wearable device that you go around and it can measure temperature, light, and pollution, air quality. All of these things are going to be available, and to get lifetime data like this, I think is going to be a profound change in the way we approach health.
how do we scale all of this? Let's say we find the right answers and the right trajectories, the right interventions. I don't think, a lot of us are doing this in a context that's, costs a lot of money right now because it has to. But which of these interventions are ultimately scalable and deliverable in a democratized way?
Well, I think many of them are going to happen. Just look at the cost of the full genome sequencing. We're down to $400 in the U.S. right now. And that's what I've seen. I've been doing longevity medicine since 2005, late 2004. And I look at the costs of the things that we were doing back then. We were spending $5,000 to $10,000 on testing that today we get for, under $200. And these early adopters that are spending the money on this are kind of pioneering the way, but they're also finding out what's working and what's not working and getting it into a cost-effective place very quickly.
Well, great. Well, thank you, Daniel. Just one last answer, like 10-word answer. I want to have you back on the show. But how well is all this going to work? Can I have you back in 5 years, 10 years, or can I have you back in 50 years? I mean, are we still going to be like doing this then?
Yeah, I think 50 years from now we can have a meeting.
All right, sounds good. Thanks, Kalisha, and thanks a lot for coming on the show, Daniel.
Thank you.
Uh, I want to remind everyone to use the chat function and panelists and all attendees option to leave comments and feedback on the show, particularly with a show like this, which is a little bit different. We can have future shows also directed more at what's going on in the clinics. If you're interested, the next Healthy Longevity webinar is August 8th next week, and it'll be Kristen Gribble, an associate scientist at the Marine Biological Laboratory in Woods Hole, Maine. And she'll be talking about the effects of maternal age on offspring health and lifespan. And to leave you tonight, I'm going to show you a video of an Australian swimmer who's breaking world records at 99 years of age. Thanks for joining us.
Boy. So he's past the halfway mark. Come on, Georgie. Come on, let's rev him up. Let's go, folks.
Thank you very much.
He's exhausted.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.